Okay, we do have these large molecules, though, remember, that couldn't get through the membrane, and we haven't really talked about how they get in and out of the cell yet. Usually they're going to be moved through what we call a vesicle, which we talked about in the cell structures presentation, or a vacuole. And so these are little membrane-bound structures that are floating around in the cell that can carry stuff in them that allows them to get out of the cell itself. I'm going to talk a little bit about how that works. The first type of movement of large molecules is called endocytosis. The cell can eat things or drink things basically through this process. I'm going to talk more about that in the next slide, so we'll just kind of hold that for a minute. But then we can also send things outside of the cell through exocytosis. That makes sense, right? Like exiting the cell. Oops. I want to talk about this picture, which is what this picture shows right here. There it goes again. Okay, so on this picture, we've got this vesicle that's got a bunch of molecules in it. Maybe this is a bunch of waste products that the cell needs to get rid of. Again, a big vesicle full of waste products. Maybe it was a lysosome that broke down something in the cell and now it needs to get rid of it. So that vesicle travels through the cytoplasm and it actually bonds with or fuses with the cell membrane and then pops open and releases all these little molecules outside of the cell. And you'll notice that the membrane of the vesicle itself here is actually becoming part of the cell membrane. So through exocytosis, the cell membrane actually gets bigger because it's adding in the membranes of these vesicles. So exocytosis, again, is a vesicle fuses with the cell membrane and releases its product to outside the cell. Now we can talk a little bit more about endocytosis, getting things into the cell. So phagocytosis, as I said in the first slide before this, is cellular eating. So you can see here we've got a big chunk of, I don't know, some sort of molecule, maybe a big protein or something right here, that actually gets kind of embedded into the cell membrane. The cell membrane wraps around it to form a little vesicle that now contains that product, that protein or whatever it happens to be. So in this process, the cell membrane is actually getting smaller because it's creating these little vesicle packets around the stuff that's coming into the cell. The second type, as I said before, cellular drinking, or pinocytosis. With pinocytosis, it's the same idea. Okay, we're wrapping part of the cell membrane around a little bit of fluid in this case and bringing it into the cell through endocytosis. So it's part of the cell membrane again, getting smaller. Could be any fluids. Okay, it doesn't have to be, targeting a specific product like phagocytosis. Maybe it's just grabbing proteins or something like that. And then the last one, it's a little more specific, it's called receptor-mediated endocytosis. And what's going on here is there's these little tiny receptors on the surface of the cell, oftentimes carbohydrates. We talked about those in the last podcast. See these little Y-shaped things? Okay, they're little receptors on the outside of the cell, and they have on them a place where, hopefully you can see, little tiny purple triangle attaches and sits in there. So that little purple triangle fits right in there. And you can see all these receptors right in here have one of those little purple triangles attached in there. Basically, when an area of the cell membrane has its receptors full of whatever molecule they bind with, then it'll start to fold in and then create a vesicle. So as it says here on the side, it's triggered by a molecular signal. In other words, the receptors have to be triggered before it actually forms a vesicle around whatever it is that it is bringing in. Whereas I said up here, this is a non-specific process. It doesn't have any receptors, so it just brings in whatever is around the cell. It's not waiting for a trigger in order to bring something in. Oh, there it goes again. Keeps jumping ahead on me. Alright, so 3 types of endocytosis. We talked about exocytosis. One more thing that I wanted to mention before we move on to our last part is that both endocytosis and exocytosis do require energy in the form of ATP as well. Okay, so it's not something that the cell just does because of diffusion, things in high and low concentration. It takes some energy to form these vesicles. So that also is, something the cell has to spend in order to have happen. Okay, I think we're ready to move on now. So we talked about large molecules. We talked about molecules going through protein channels. We talked about molecules that can go between the lipids. Our last category is, well, what about water? And I promise you that I talk more about osmosis, and this is the point at which we do that. So osmosis is specifically the diffusion of water, and water is really important to your cells. And so we have a very special case of diffusion where we can spend a lot of time talking about how this works in the form of osmosis. Again, it's still diffusion, so it's going from high to low concentration, right? High to low concentration, but what we have to track the concentration of is the water. So in other words, it's always going to move water from where there's a lot of water to where there's less water in an attempt to balance the concentration of water on both sides of the cell. So diffusion has to be water. That's our number one part. And then number two, we have to have a membrane. We have to have something to move the water through that controls or regulates how much water is moving. We've got those two things, the water and the membrane. We've got osmosis. So let's take a little bit more close look at osmosis and what happens. We have two different types of situations that we have names for when we're talking about the concentration of water. So if we have very little water in an area, so if a cell doesn't have a lot of water, or if the outside of the cell doesn't have a lot, or whatever the area is, where there is less water, we call that area hypertonic. Right? So less water is hypertonic. How do we get less water in an area? Well, there's more stuff dissolved in that area if there's less water. Okay, remember our words that we've talked about before, 10th grade biology for sure, and at 9th grade physical science I believe as well. We've got the solute and the solvent. Okay, the solute is the stuff that's dissolved in the water. The water is the solvent. So if we've got a lot of stuff dissolved, okay, we don't have very much water. And so we call that, again, hypertonic. Okay, so that's our first condition. You can see that over here on this side of the membrane. Lots of stuff dissolved, right? That's the yellow stuff. That's the solute. Which means if we have a lot of solute on that side, there's not as much space for water, so that's going to be hypertonic. The other osmotic condition that can exist is called hypotonic. And in a hypotonic condition, we have lots of water. So, lots of water, hypotonic. Sometimes it's easy to remember H2O and the hypotonic. So lots of H2O is hypotonic. What does that mean for the solute then? That means we have less solute dissolved in it. So this side over here of our membrane is hypotonic. Lots of water, less solute. Again, the solute is the little yellow dots. Now, if we were to even out the amount of water on each side, we would call our situation or a condition isotonic. Equal amount of solute and water on both sides. Now that doesn't necessarily mean the molecules, like in this example, the yellow molecules would be even, because we're going to assume those yellow molecules can't move. They're stuck where they are on one side of the membrane. But we could move water back and forth. For example, we could move water from the hypotonic area where there's lots of water to the hypertonic area where there's less water and dilute this area a little bit so it's not as concentrated. And then we would be in a situation that we'd call isotonic. So the concentrations are the same on both sides. Not necessarily the number of molecules, but the concentration of the molecules are the same on both sides. So this little diagram here down at the bottom to finish that up shows that our water always moves from an area where it's hypotonic to an area where it's hypertonic. Because hypotonic, remember, has more water. Hypertonic has less water. And osmosis is the movement of water from where there's more to where there's less. So always move from hypo to hypertonic.